everybody. Welcome back to Environmental Organic Chemistry with Lisa. We're going to continue to talk about hydrolysis. I know this is a long topic with lots of little subsections. We're going to have a subsection here about the hydrolysis of our carboxylic acid and carbonic acid derivatives. And this is an important topic because these chemicals are important in all kinds of environmental situations. So, for example, here's endosulfan. That's an important environmental chemical. Malathion is one of the most widely used organophosphorus pesticides. Aldicarb is a carbamate pesticide. Here's this benzyl butyl phthalate thing. So phthalates in general have this structure with the two ester functional groups here. And so all of these chemicals are environmentally relevant, and they can hydrolyze by the same set of mechanisms. So what happens is you have this sort of structure here where you have L. Remember, L designates a leaving group. And then you have some sort of chemical, some atom double bonded to another atom. So like in this case, it's sulfur double bonded to oxygen. Here in malathion, it's phosphorus double bonded to sulfur. In aldicarb, you've got the carbon double bonded to the oxygen here. Same thing going on in these phthalates. So you've got this structure, and then this thing from here over can be the leaving group. Same thing here. You can actually have several leaving groups. This and these also, these methanol derivatives can be leaving groups. Here, these would be leaving groups. And carbamate derivatives have a very complicated hydrolysis where this whole thing splits off as CO2, and then both sides of the molecule are considered leaving groups. So you have, again, this double bonded structure with a leaving group on it. And what happens is that hydroxide or the nucleophile can come in and attack here because all of these bonds involve some sort of polar interaction. That's most easily seen here with the carboxylic acid or ester groups, that the CO bond is a polarized bond. Carbon has a partial positive charge, so the hydroxide is going to be attracted to that, comes in, pushes the electrons up here so that you have a negative charge, a formal negative charge here. And then when that negative charge swings back in to reform the double bond, it results in the leaving group leaving. So this is the mechanism by which you can hydrolyze carboxylic acids and carbonic acid derivatives. And notice that when we have this structure with the double bond here, we have a carbon that is not aliphatic. So we just spent a lot of time talking about SN1 and SN2 reactions. Those only occur for aliphatic carbons, carbons that are sp3 hybridized that have bonds to 4 other atoms. But here we have a carbon that is not sp3 hybridized. It is now sp2 hybridized, has the double bond here, and it is still susceptible to hydrolysis, but it has to have this very particular kind of structure. So in this structure, the Z can be carbon, can be phosphorus, can be sulfur. The X up here can be, usually is oxygen, but could also be sulfur or an amine group. And the leaving group, could be anything that's a good leaving group. So it could be halogens, it could be sulfur species, it could be amines, it could be oxygenated things. So all of these can serve as reasonably good reasonably good leaving groups. So this is just some terminology in case you've forgotten. I'm sure you have. I couldn't tell you what a carbamate is or a sulfonic acid ester. So if you have any questions about that, you can look at some of the terminology just to remind you what some of these functional groups might look like. And I guarantee you, as I do every year, the final exam will have some of these types of structures on it. And I won't ask you to name them, but I will want you to be able to recognize that those are structures that could undergo this type of hydrolysis. So there's 3 mechanisms by which this can occur: the neutral, the acid catalyzed, and the base catalyzed. So the neutral mechanism is going to occur at relatively neutral pH, and as a result, the nucleophile is going to be the water molecule. So water molecule comes in here, attacks at the carbon, the negative charge goes up there, and then pretty quickly you get a proton transfer. This proton moves from here up to there. It actually could be a completely different proton. Doesn't matter where the proton comes from, but you get this structure where both, sort of both ends are protonated now. And you can also get a proton transfer over here to the leaving group, and the leaving group could leave as a protonated leaving group. And that is important. If the negatively charged leaving group is very, very unstable, it might be much happier to leave as a neutral compound. Either way, you get some protons moving around and you get the leaving group gone. This can lose its proton, or you can go this route, and either way you end up with carboxylic acid here as your product and the leaving group. Okay, so the way that you know that this is neutral is by the fact that water is the nucleophile, and you end up with neutral chemicals here. There's just enough protons to go around, which is how you know it's occurring at a neutral pH. So this neutral mechanism is most likely for when you have, occurs most commonly when you have good leaving groups. If your leaving groups are poor, you might need a different mechanism to get this reaction to go. The acid catalyzed mechanism is going to occur, under acid conditions. Under acid conditions, you don't have a lot of hydroxide around. What you have is a lot of H3O plus and a lot of water molecules. So this hydronium, H3O plus, this is not a very good nucleophile, right? It's not negatively charged, and it only has one set of lone pair electrons now because that extra hydrogen has consumed one of the sets of lone pair electrons on the water molecule. So it's really not the greatest nucleophile in the world, and so you can see it doesn't actually act as a nucleophile. It doesn't come in and attack at the carbon. What it does is it just protonates the oxygen up here and adds a proton here. Okay, and that sort of activates the backbone, and now this whole thing has a formal positive charge, and that makes it very attractive to a nucleophile, even a relatively weak nucleophile like water. So now water will come in and attack here at the carbon. So there's where you get your water molecule right here, and we're under acid conditions, and so you have plenty of protons to go around, so this remains protonated, and then you can get a transfer of the proton over here to the leaving group. That makes this a better leaving group, and it will leave as the protonated and therefore neutral species. And again, there's plenty of protons around, so it will leave behind the protonated carboxylic acid. So this tends to be important when there are no electron withdrawing groups and the leaving group is pretty poor. So by protonating the leaving group, you can make it a better leaving group, and that's why this mechanism is most likely to occur when you have a poor leaving group. And one of the key factors is how strong a base is this ester functional group. How good is this functional group at attacking or attracting this proton? And that will determine how many of these molecules end up in the protonated form, and the protonated form is the one that goes on to react via the rest of these mechanisms. So that's the key question in determining how fast this reaction could go forward. The base catalyzed mechanism makes a lot more sense because we're under base conditions. We have hydroxide around. Hydroxide's a better nucleophile, so it acts as the nucleophile, comes in and attacks. When it does, the electrons kick up here, but there's not enough protons around to protonate this thing, right? We're under base conditions. We don't have enough protons around, so this will stay negatively charged. And as a result, the leaving group will have to leave as a negatively charged species. There's not enough protons around for it to be protonated. It could, after the fact, capture this proton from the carboxylic acid, and if it's a stronger acid, it might pull that proton away, but you're still left with the negatively charged carboxylic acid group. So this tends to be a more common mechanism when you have a good leaving group. So the base catalyzed mechanism is going to be more important when the leaving group is pretty stable and can leave pretty easily. So what you get is this kind of strange situation where the reaction rate, and in this case it's expressed as the half-life. Don't know why I get those little spikes every time I do this. But anyway, the half-life is a complicated function of pH, because over here you have the base catalyzed mechanisms, and then over here you have the acid catalyzed mechanisms, and in the middle you might have some neutral mechanism. I'm doing a bad job with that, but here's the neutral mechanism. And so you get this kind of weird situation where you have kind of a three-phase distribution. You've got some acid catalyzed mechanism going on over here. Here it's the neutral mechanism, and down here it's the base catalyzed mechanism. So The pH dependence of the rate of these reactions can be very complex. It depends on the structure of the chemical. That's all we're going to say about that for now, but the next lecture is going to talk about how you would predict the rates of some of these reactions.